This will be Module 2 of our NAS2 review, and this module covers energy. And again, this is meant as a supplement to the readings you should be doing in your Canvas course shell. Energy basics here. Potential energy is stored energy, and for our purposes, what we're going to look at a lot is the energy stored within chemical bonds. You put energy into making a bond. When you break a bond, that energy gets released. The other place we'll look at when we talk about potential energy is the concept of concentration gradients. For instance, if you had a lot of, sodium, a lot of sodium on one side of a cell membrane and not a lot on the other, that's a concentration gradient. When you see that word gradient, just think of a difference. If you were to suddenly allow the sodium to cross that cell membrane, going down its concentration gradient from high concentration to low concentration, that energy would then get released. Kinetic energy is energy in motion, so that would include radiant, sound, mechanical, light. That's all kinetic energy. I put a picture down here of a dam. I like to use this illustration because if we look at the water behind the dam, that's potential energy. If we look at the water coming over the dam, that's kinetic energy. Chemical reactions. There are reactants and they form products. In the reaction I've written here, we have A and B. Those are the reactants. They form the product AB. If you notice this arrow goes in one direction, that means that that chemical reaction can only go in one direction. Some reactions are what we call reversible, and you'll see the arrow pointed in both ways. We saw that when we looked at the carbonic acid buffer in the last module. The reaction rate is how fast that reaction happens. And we'll look in a minute at enzymes, which are catalysts, which can increase the rate of the reaction. Synthesis reactions in general are going to require energy. When we put energy into a reaction, we call that an endergonic. Synthesis reactions are referred to as anabolism. Degradation reactions, by contrast, generally release energy, and we call those exergonic reactions. They are a type of catabolism. The word metabolism includes all the reactions within the body. So it includes all the anabolic reactions and all the catabolic reactions. This is an example of a dehydration synthesis. So we're making something here. And it's called dehydration because we're going to take an oxygen and 2 hydrogen out. And as a product, we're going to get water. So we're dehydrating it. And then we have a bond here. Now, these are two monomers. These are two monosaccharide monomers being bonded together to form a what we call a disaccharide. This is a dehydration synthesis. This would be endergonic and an example of anabolism. In contrast, this is hydrolysis. This is a disaccharide, two monosaccharides put together, and we're going to add water to this, and it is going to break this bond here and form two monosaccharides. So this is an example of a catabolic reaction. It would be exergonic because when you break bonds in general, energy is released. Enzymes. In human systems, enzymes are going to be proteins. Remember that proteins have a specific structure and they can be denatured either by an increase in temperature or any change in pH. Enzymes are specific for a specific reaction. And typically we're going to see reactions occurring one after another to form what we call a metabolic pathway. And each of those reactions would have a different enzyme. The substrate is what is going to bind to the active site of an enzyme. And it does this in what we call an induced fit model. So whatever the substrate is, it could be a disaccharide that's going to be hydrolyzed, for instance. It will come into the active site, and that enzyme is going to kind of wrap itself around it, sort of like a hug. Now, enzymes can be inhibited. An inhibitor could bind to the active site and not allow that substrate to come in. The reaction rate, now enzymes increase reaction rate. What they do is they lower the energy the amount of energy that needs to be put into a reaction to get it started. So an enzyme can increase the reaction rate. If you increase the enzyme, you increase the rate. Other things that can increase the rate, well, putting in more substrate. You can increase the temperature. Now, gotta be careful here. If you increase temperature just a little bit, it actually increases the reaction rate. A slight increase in temperature moves molecules around more. Substrate makes it easier to find the enzyme. However, a higher increase in temperature is going to denature the enzyme. pH. So certain enzymes work at certain pH. Again, if you change the pH too much, you can denature the enzyme. And then inhibitors. And as I said before, an inhibitor can bind to that active site and not let the substrate bind there. Also, inhibitors can bind somewhere else on the enzyme and change the shape of the active site so that a substrate cannot bind. This is just an example of an enzyme. The enzyme is the dark gray blob here. It has an active site. Here's a substrate coming in. So the substrate comes in. The enzyme is going to kind of hug it and form this enzyme substrate complex. Chemical reaction occurs for this particular reaction. This is catabolic. We've got what looks like a dimer being made into two different monomers here, the products. Now the enzyme itself is going to come back and can do that same reaction over again. So the enzyme doesn't get used up. It winds up being the same as it was when it entered the reaction. ATP and cellular respiration. Remember ATP, adenosine triphosphate. There are 3 phosphate groups on this molecule. The bond between the second and third group is a high energy bond. When you break that bond, energy gets released. ATP is the form of energy our cells need. Now, you may think, well, food is a form of energy, and we eat food, it gets broken down into, for instance, glucose. But glucose is not the form of energy our cells need. Cells have to change the glucose and take the energy from that and put it onto ATP in order to use that energy. What happens is ADP, which has 2 phosphates, adenosine diphosphate, uses energy and gets another phosphate group added to it to make the ATP. So again, general concept, when you make a bond, it takes energy. If you break a bond, it gives off energy. To make the most ATP, our cells need oxygen. And it does a process called aerobic cellular respiration. It's also referred to as oxidative phosphorylation, and it occurs in the mitochondria. I'm going to do just a quick review of cellular respiration. What you see here depicted in this kind of an orange here, this is a mitochondria. We all learned that the mitochondria is the powerhouse of the cell, but no one usually can tell me why that is. What we're going to start with is some sort of carbohydrate, and this is going to occur in the cytosol, which is the liquid portion of the cell outside the mitochondria. That carbohydrate is going to get broken down and form glucose, which is a monomer, and that glucose goes through a process called glycolysis. Glycolysis occurs in the cytosol, and you get pyruvate. Pyruvate is a product of what we call glycolysis. Now, pyruvate can have 2 different fates here. One is lactic acid. If no oxygen is available, pyruvate gets turned into lactic acid. If you've ever run or ridden a bike, let's say up a hill, and your muscles start burning, that's because you're not getting enough oxygen to the tissues, to the muscle tissue, and you're forming lots and lots of lactic acid, which burns. Now, the process of glycolysis produces some, up to ATP, but not a lot. If oxygen is present, that pyruvate molecule is going to go into the mitochondria. It's going to be turned into something called acetyl-CoA, and it's going to go through the citric acid cycle. Now, we'll go into much more detail during the semester when we get to that chapter. During the citric acid cycle, different bonds are broken. Remember, breaking a bond releases energy. When you break those bonds, electrons get released. Okay, electrons get released. And these electrons have high energy attached to them. And then they're going to go through something called the electron transport system or electron transport chain. And oxygen, this is where the oxygen comes in. Those electrons go through this electron transport system. And as they do that, they release energy. And that energy forms ATP. At the end of this system, oxygen takes those electrons and water gets formed. Right? When oxygen is present, lots and lots of ATP gets made, 38 plus. Now, glucose isn't the only thing that can go through this process. You can also take fats. Remember, the triglycerides are our way of storing fats, and fats are energy, so it's our way of storing energy. Fats get broken down to fatty acids, and they get broken down into acetyl-CoA, and then they can go through this system to make lots and lots of ATP. Same thing with proteins. Proteins can get broken down to amino acids. Remember, that's the monomer, and they can go through this system. Although in general, our cells don't break down proteins because proteins are so important. There are enzymes, they are our cell structure. So it's only in extreme starvation that a protein would get broken down. So just to review, in the cytosol, we have a process called glycolysis, and glucose gets converted to pyruvate, and you get only 2 ATP out of that. Pyruvate can then, in the presence of oxygen, go into the mitochondria and form acetyl-CoA, which goes through the citric acid cycle. Different bonds get broken. Electrons come off of that. These have lots of energy in them because that was the energy in the bonds. The electrons go through the electron transport system. All that energy gets released, and you get about 38 ATP. Electrons eventually join with oxygen to form water. If no oxygen is present, that pyruvate back here, pyruvate gets turned into lactic acid, and that's why your muscles burn when you do a lot of exercise. This process is called the oxidation of glucose because you're adding oxygen to glucose. And I've written it out in words here since we haven't learned a lot about the chemical formulas and everything. But glucose plus oxygen, those are the reactants. And this arrow represents glycolysis, the citric acid cycle, the electron transport system, all of those different pathways. And there are multiple enzymes involved here. The products are water and carbon dioxide, plus you get lots and lots of ATP. Glycolysis alone makes a net of 2 ATP, whereas if you have oxygen present, you get 38, could be more. You also can use other reactants, for instance, fatty acids or amino acids. They go through a similar process and you get lots of ATP. Now, how do you get the ATP? Well, as you break the bonds in the glucose and all of the intermediate molecules, energy gets released. That energy that gets released is going to be used to take a phosphate group, that's a phosphate group, and add it to ADP. ADP is adenosine diphosphate. There's only 2 phosphates here. So you can add a third one, but that's going to take lots of energy to add that. So you got to put energy into this part of the equation to make the ATP. But now that you have ATP, the cell can use the ATP by releasing breaking that final bond, releasing the energy. That energy can now be used to do different cellular functions. And you wind up again with ADP, but immediately a phosphate group gets added and you get more ATP. As long as oxygen is present and you're able to do that oxidative phosphorylation. And that's it for the second module, energy.